Okay, so I'm starting. If I'm not live, somebody will stop me. So good morning, United States. Good afternoon, Europe. And hello to everyone else in the world. You're at least as important as we are, but morning and afternoon is all I can do now. My name is Carsten Schradin. It's a pleasure for me to host today's FINE seminar. I'm myself based in Strasbourg in France. But before we come to today's talk, I would like to quickly look back at what we did last week when Vanessa Esenwa was talking about sickness and health, complex effects of social behavior and infectious disease. A really fascinating talk, and I love chatting to her afterwards. Disease, transmission of disease, surely an importance for everyone working with social animals. And if you have, for whatever reason, missed this talk, you can go on our YouTube page and look at the recording that we have here. And already, as not everybody can stay until the end of the discussion, looking forward to next week. I will unfortunately miss it as I will be in Namibia for a conference. There will be Peter Baston talking about social evolution in anemone fishes formation, maintenance and transformation of social groups. But today we have a talk by Mark Harrison from the University of Münster in Westphalia, Germany, talking about one of my privately favorite animal groups. This are the Platodea, the cockroaches, and termites, and he's going to talk about molecular signatures of longevity in eusocial animals. He will make his talk a bit broader. So Mark, he studied in Germany, getting his master's in 2012 at the University of Bochum in biodiversity. Then he went to the University of Leicester in the UK for his PhD, where he worked on bumblebees, eusociality and genomic evolution. So he is a specialist not only for social behavior, but also for genomics, which of course is very interesting. I mean, when we talk about evolution in the end, I think we all agree you must somehow see it somewhere within the genes. And I'm sure we're going to hear quite a bit about this today. And then in 2018, he came to the University of Münster, where he is currently first as a postdoc. And since 2016, he came to 2016, he came to Münster as a postdoc for 2 years. And since 2018, he's there a lecturer. But unfortunately, like it's one of the diseases of the German system, the lecturers, there are not permanent positions before you become a full professor, which normally you cannot become at the university where you are at the moment. So he is not tenured yet, which is the mistake of the German system, but he is doing great research. He has published a very interesting paper in Nature Ecology and Evolution about the evolution of eusociality. He has lots of paper in molecular ecology and in journals on genomics, and is doing very interesting talk and work. And yeah, I'm very much looking forward to your talk now, Mark.
Thank you for the introduction. Hello, everyone. Right, the obligatory question, can everyone see the presentation? Yeah, okay. And can you still see it in the correct mode? Yeah, good, okay. So I'm sorry, I've slightly changed the title, but nonetheless, it's the same topic, genomics of healthy aging in eusocial animals. And it's true, I've done quite a bit on Platodea, and I will be talking about Platodea a bit, but I will also extend into other eusocial groups. So first of all, eusociality, there are several definitions and different levels or different aspects to this complex trait, but I think maybe one of the most important parts of this complex trait is the division of reproductive labor among castes. Here we have a section of a termite colony, and here we have the fertile queen, and then the sterile workers and soldiers. So it's important that they don't differ by genotype. They share a genome, but you have these very, very distinct phenotypes within the same colony. And what has fascinated me, one of the reasons that's brought me to eusociality is that it's evolved convergently several times in very different groups. So possibly around about 170 years ago in Blattodea, the termites evolved from within the cockroaches. And then it's not quite clear when, but around about the same time in Hymenoptera, several groups have evolved eusociality, a couple within the bees. The ants have one origin of eusociality. There are possibly two origins of eusociality in wasps. Even in the beetles, there is at least one species which could be defined as eusocial. That's the ambrosia beetle, Ostroplatipus incompertus. And even if we look outside of the insects, we can consider the naked mole rat also as eusocial, right, within the rodents. Another fascinating aspect of eusociality is is when we consider the trade-off between fecundity and lifespan. I mean, generally among animals, you see this trade-off: either an animal invests more in fecundity or more in lifespan, right? So a mouse, for instance, will just live a couple of years, but will produce several litters per year and several pups per litter, maybe 30 to 50 pups per year. Whereas an elephant will live for decades and likely only produce a single offspring every 4 to 5 years. And we see this throughout the animal kingdom. And in experiments where people have tried to manipulate these two aspects of the trade-off, as always, as most in Drosophila, for instance, most often leads to a trade-off, right? So reducing fecundity or postponing fecundity to later in life will lead to an increase in lifespan in flies. Well, this trade-off doesn't seem to apply to eusocial animals. So some higher termite queens can live for 30 years while laying thousands of eggs every single day for their whole life, and don't seem to show any signs of aging. Whereas closely related species live much shorter. I mean, it's quite interesting that the sister group here, the wood roaches, are subsocial and they live for 7 years. That's already pretty long for an insect. And the German cockroach, for instance, is known to live around 100 days. It's also quite long for an insect, but as you can see, a completely different level to the termites. And incredibly, this has happened several times in each of the groups we discussed on the previous slide, where they can live. Eusocial queens can live for decades, whereas the non-social relatives just live weeks in Hymenoptera or years in just a few years within the rodents. And I've sort of made it my mission, and since moving to Münster, to try and understand the molecular signatures of this, starting off with what are the molecular signatures of the multiple origins of eusociality. And as Carsten already mentioned, this started off with looking at the evolution of termites. And these were the genomes that we were working with back then. We had some new termite and cockroach genomes, which you can see at the bottom. Bija is the German cockroach, is an outgroup. And then we have three termite genomes, and we tried to look for molecular and genomic signatures of eusociality within Blattodea, and to see how they compare to previous findings for the evolution of eusociality in Hymenoptera, which we see in the blue branches. So this is a PCA, a principal components analysis, showing the first 3 principal components, which explain roughly 70% of the variation in methylation patterns. So I looked at predicted methylation, which you can do from CPG patterns within the genome, and estimated this across all of the genes in each of these species I was looking at, and then compared the patterns, so the distribution of these predicted methylation levels across all of the genes within a genome among these species. And what's striking here is that the termites cluster together very, very strongly, and quite different from all of the other species we looked at. So there seems to have been a strong change in methylation patterns in termites compared to the other insects we were looking at. We also found evolutionary changes in zinc finger transcription factor families. And these transcription factors were enriched for queen-biased expression, meaning that they were important for the regulation of castes, and possibly the evolution of these zinc finger families, possibly important for the evolution of castes in termites. There are similar findings before then and also since then in Hymenopteran lineages, finding major changes in methylation and transcription factor binding sites in ants and bees. And this all makes sense, right? Because with the evolution of eusociality, the first major step is major changes in expression patterns, leading to distinct expression patterns among the at least two castes within a colony. So it's an increase in transcriptional, the complexity of transcriptional regulation, which we see evolving at all origins of eusociality, at least in insects. Furthermore, we found interesting patterns in the evolution of chemoreceptors, namely inotropic receptors. These have this structure with the lobe here, which is important for detecting chemical cues. These chemoreceptors were massively expanded in cockroaches and also in the termites. And interestingly, a different chemoreceptor is known to be expanded in the Hymenoptera. And this is the kind of thing, what we were actually expected to see expansions in these odorant receptors in the termites. That would have been a really cool finding. Oh, look, they're expanded here as well. And they've been known to be important for the evolution of sophisticated colony communication within the Hymenoptera. It's important for nest mate recognition, for instance. The odorant receptors were not very abundant at all in Blattodea. In fact, the inotropic receptors, like I say, were highly abundant. And so this is a case of convergent evolution, because we found plenty of pieces of evidence that show that the most likely important for caste-specific roles. So we detected caste-specific expression of these inotropic receptors and evidence for positive evolution, positive selection within the binding lobe, which indicates an adaptation to different chemical cues that could be important for foraging, but also, like I say, chemical communication within the colony. And there were some other findings, but I'd like to move on to the next topic that I was interested in trying to understand, and that is the reversal of this fecundity longevity lives trade-off, in eusocial insects, and see if I can find a molecular signatures of that. So I started off by comparing. So I had transcriptional data, so RNA-seq data for old and young ant queens of this species, Cardiocondyla obscuria, and I took those data and developed a co-expression network. So co-expression network depicts the correlation or the co-expression between genes. Right? So how closely co-expressed genes are among different conditions, in this case among individuals. And each dot in this network, this is represented in two-dimensional space here. So each dot here, each node represents a gene, and the connections between those genes represent the correlation in expression, so the strength of the co-expression between those genes. And the closer, so the shorter the edges, the closer the nodes are together, the more strongly co-regulated or co-expressed they are. And then the assumption is that they're also strongly co-regulated. And these are colored by modules. Each module represents a group of genes which are particularly strongly co-expressed. And I correlated the expression of the genes within these modules with the trait, and the trait of interest here is age. So we had old and young queens, and I had a look which of these modules were significantly correlated with old queen expression and with young queen expression. And all of the old bias modules clumped together in one large cluster here, which is circled in blue, and the young biased modules were all grouped together in one large cluster in the young biased, all the young bias modules grouped together in this one cluster over here in the green circle. And I had a look at what functions were enriched within these age biased, these old biased modules. And a lot of these could be related to hallmarks of aging or mechanisms to counteract hallmarks of aging. For instance, the maintenance of proteostasis seemed to be enriched among these modules, or transcriptional regulation and stress response. So proteostasis and transcriptional regulation, they're two of the major hallmarks of aging, right? These are the two of the things which break down with age, and it appears that they are really well regulated in old age, possibly explaining how they manage to remain highly fertile at old age. Next, I was wondering if there would be a selection shadow. So a selection shadow has been put forward as one of the theories of evolutionary theories of aging. The idea is that genes which are expressed or are important for survival at old age, selection is less efficient on them because they're expressed in fewer individuals. So here, if this is a survival curve here, the effective population size decreases with age, so the efficiency of selection decreases with age. And looking at data for Drosophila, we do indeed find an increase in DNDS in old biased genes, which indicates weaker purifying selection. So a DNDS closer to 0 indicates strong purifying selection. And there is a significant difference between genes expressed at young and at old age. And this could indicate a selection shadow, so weaker purifying selection acting on genes at older age. And when I had a look in the ant queens, we didn't see this. In fact, we saw the reverse, which is really cool. So it appears that the genes which are highly expressed in old queens are actually under stronger purifying selection. So no selection shadow. And this could be related to a decrease in extrinsic mortality. So the queens are well protected in the nest. So we could see them going along this red curve. This red line here in the graph at the bottom indicates survival rate in captivity. So there does not appear to be a selection shadow, which could also explain why genes are well regulated at old age and well functioning, because they're under efficient selection. In another study, we had some transcriptomic data for the higher termite macrotermite natalensis, and I also created a co-expression network here. But this time we didn't just have old and young queen data, but also king and worker data. So RNA-seq for each of these. And for instance, there was one module within there which was strongly correlated to worker expression. But again, there were modules related to these were parts of the network which were activated in young queens, and other parts which were activated, so overexpressed in old queens. And when I compared between these, so I had different methods to try and see if I can find overlaps between modules, which are so young biased and old biased modules. I only really found overlaps in the old biased modules, which is really cool. So we found several functions which were enriched there. So it wasn't the same genes, and it's difficult to find orthologs anyway between these two species, but go-term enrichments were similar, and they had a significant overlap between these. And also some preservation statistics showed that there were some structures within those modules which were strongly preserved. Preserved is possibly not the right word here, but convergently involved in each of these lineages. Yeah, and my colleague is currently working on trying to find out whether we can find these kinds of patterns in several eusocial lineages. He's comparing co-expression networks among different lineages, including bees, ants, termites, and also non-social lineages like Drosophila. Right, another thing I looked at with these co-expression networks is how connectivity, so the correlation among genes, how that changes with age. And then connectivity could be seen as a proxy for transcriptional regulation. So the more strongly correlated genes are, you would expect the regulation to be tighter. And this was found to decline with age in mouse brains. So when they did looked at co-expression networks in young mouse brains and in old mouse brains, they saw the connectivity decrease significantly with age. And I had a look whether we saw the same thing in the termites and the ant queens, and we did not, which is really cool. So there was no decline, and there was a slight increase, in fact, in connectivity, indicating that expression remains strongly and tightly regulated even at old age. And then when we look at specific pathways, I mean, the pathways which always spring to mind when we're talking about the longevity-fecundity trade-off are the nutrient sensing pathways, IIS, so for insulin and TOR pathways, so amino acid sensing pathway. This is where the trade-off is often measured and also experimented on in model organisms and manipulated. And one of the central transcription factors within here is TOR, which you can see in the green box, and the main upstream activator of TOR is the gene called RHEP. And we found a negative correlation in expression between them, which, okay, it's RNA, it's mRNA and not proteins, but it could be an indication that it's in an inhibitor in ants rather than an activator. And just to check whether we would also see positive correlation in Drosophila, we also had a look at the expression there. So the expression correlates positively between these two genes, as you would expect in Drosophila, and it's probably what you'd expect in other model organisms. So these pathways are highly conserved across different model organisms in C. elegans and in the mouse and in Drosophila. And in all of these, RHEB is a positive regulator of TOR, and we found a negative correlation here in the ant, but also in the termite. I realize it's still only anecdotal now, and it's only mRNA, but this is something we're looking into further. And we started off by doing some RNAi in Drosophila and RHEB. And in the near future, we would also like to test that in at least one ant and termite to see what happens if we knock down RHEB. Other proteins which are important within this pathway, the insulin-like peptides, they're important for activating the insulin signaling pathway, and upregulation of these generally leads to a reduction in lifespan, so an overexpression. And in the termite paper, termite study, we found a massive increase of one of these insulin-like peptides with age. So we have the founding queen here, so 0 months old, and then 3 months, 9 months, 31 months, and then the 20-year plus queens. And there we saw a massive upregulation of this protein. This likely allows them to increase reproduction in age. But for some reason, there are no detrimental effects of this increase in insulin signaling on their health. And this is something we're looking into at the moment. My collaborator Mireille Vasseur-Cornier has developed fat body cell cultures from old termite queens, and she's doing some RNAi experiments, trying to knock down ILP9 and see what effect that has. So that's work in progress. Right, so we didn't only look at transcriptional regulation, we also got some methylation data together. Another hallmark of aging is a breakdown in epigenetic patterns. So something called epigenetic drift is where DNA marks tend to decrease or increase more randomly with age. We didn't find that in the termite. In fact, if we compare young queens to old queens, we found an increase in the number of sites which were differentially methylated between the old queens and other castes. And this was stable across all of the replicates, indicating well-regulated, strong changes in methylation in the old queens, possibly allowing an increase in reproduction. Furthermore, another indication of how they managed to remain healthy is that we found within, so the same data set, but now the RNA-seq data again. Here we see the proportions of mRNA, which are transposable elements or repeat elements. And this decreases drastically with age in the queens. So here in the founding queens or in the workers, it's around about 20 to 25% of all mRNAs which were extracted from the fat bodies were made up by repeat elements. And it's only around about 3% in the mature queens. And this is important because transposable elements are damaging, right? They cause damaging mutations to the genome. And also, again, one of the hallmarks of aging. And moreover, the mature queens have a large increase in expression in transcription with age, which coincides with the increase in reproduction. And we found that gene expression correlated quite strongly with TE expression. So basically that means that the TEs are just being, they're using the machinery, the transcription machinery of the genes, and every time a gene is expressed, it gets co-expressed. So there's a strong correlation. But the queens managed to somehow strongly upregulate gene expression, but at the same time control the regulation of T, so they're not also strongly co-expressed and causing damage to the genome. And that is regulated by an increase in expression of members of the piRNA pathway, the pathway which is known to be important for regulating transposable elements. Right, so I've got these findings on some molecular mechanisms in two species. And then I more recently, I've been looking more towards, again, trying to find convergent molecular signatures across eusocial genomes and also outside of insects. And I think we should be able to expect convergent signatures. First of all, sociality itself, with increasing social complexity, you have a decrease in effective population size. So an increase in social complexity, in eusocial complexity, means an increase in colony size, but also a reduction in the relative number of reproducing individuals. So a large colony will be closer to 100% sterile, right? The larger it gets, the more workers you have. And so a decrease in effective population size is likely to lead to a reduction in selection efficiency, so a relaxation of selection in eusocial lineages. This is something that all eusocial lineages should have in common. And this is something we can detect in the genomes. So I had a look at, again, at DNDS. DNDS is it's like a measure for protein evolutionary rate, if you like. The closer this is to zero, the stronger the purifying selection. It's oversimplified. We did other supporting analyses as well. But with increasing social complexity as we go up here, here's where we have the termites and the most socially complex termites we have at the top. We do indeed find an increase in DNDS as we would expect, indicating a relaxation of selection. And if we also do some additional analyses, specifically trying to detect a relaxation of selection, I did this across all orthologs on these two branches. On this one branch here, the origin of eusociality, and then again on the origin of worker sterility. I mentioned earlier that some lower termites have totipotent workers that can also become reproductive later in life. But the top three termites in this tree, they all have truly sterile workers. So there we would expect the effect of a decreasing effective population size to have an effect on, to be stronger, the effect of that on relaxation of selection. And that's what we indeed find. So this is an indication. So what you do is you compare the evolutionary rates on that branch against other branches in the tree. And an intensification of selection would be seen in a value greater than 0 and a relaxation lower than 0. And this is the distribution of those. And we see that the majority of genes have a relaxation of selection along that branch, which would lead to these higher DNDS values. And I find the same for naked mole rats. So here we have NMR, that's the naked mole rat, and the Damaraland mole rats, also eusocial, not quite as socially complex, and then some related less social rodents. And here again, we see an increasing DNDS and a relaxation with in the naked mole rat branch. And I propose this is due to a decrease in effective population size due to eusociality. And there are similar results, published results. Sorry, I forgot to put a citation in here, but there are at least 2 studies I know of where this has been found in Hymenoptera. So it seems to be widespread across eusocial lineages that sociality, in this aspect at least, can shape genomes. But I also have some other ideas of why and how eusociality should shape genomes and should be detectable in convergent signatures. In a highly eusocial organism, a genome encodes for at least 2 very distinct phenotypes, right? So you have the reproductive queen or king, and then the sterile workers and soldiers. So on the one hand, you have this direct selection working on genes which are important within the reproductive individual, because that can be passed on directly to the offspring, and indirect via the fitness of the colony selection on the sterile individuals. But apart from that, you also have different selection regimes acting on these, right? So they're very, very different behaviors and physiologies of these, whereas the queen is generally well protected from dangers, from selection. The workers are much more exposed to different selection pressures. So we would expect to see different levels of selection within the genome, depending on whether a gene is more strongly expressed in a queen or in a worker. And this is something we should see convergently among eusocial lineages. So for example, if we have 2 mock eusocial genomes here and 1 non-eusocial genome, we have different colors for depicting whether a gene is overexpressed in one or the other caste or not differentially expressed. So the black genes here are not differentially expressed between castes. The red genes, or brown, or depending on whatever color you see it as, for instance, queen bias. So they're overexpressed in queens and hardly expressed in workers, and the greens, the opposite. They're strongly expressed in workers. Very simplified model. So based on the ideas from the slide before, not just my ideas, of course, I would expect strong differences in selection between a worker and a queen gene within one genome. So a divergence there in selection, but a convergence within a caste between species. So we would likely see similar selection patterns happening on a queen gene in a termite and in an ant, possibly more similar than the selection acting upon worker and queen genes within one genome. So that's the selection acting on the genome, and we can find signatures of selection in the genome. And then there's also the problem of the regulation. So each of these arrows depict transcription. So these genes need to be differentially regulated in very different individuals, different groups of individuals within a colony. So there's that complex regulation should also be visible within the genome. Well, the question is, how do we go about finding these patterns? I mean, we've tried doing comparative genomics and we see some interesting comparisons, but it's really difficult to find these signatures just by comparing the genomes between Hymenoptera and Blattidaean genomes, for example. First of all, it's really difficult to find orthologs, right? And if we look at this, we're also likely to see signatures outside of genes. So I'm not only interested in genes, I'm also interested in non-coding regions, in regulatory regions. So I thought, okay, let's try some new tools. Let's have a go with machine learning. Is it possible to detect these molecular signatures of eusociality with machine learning? So I took a multi-layer perceptron, which has these several hidden layers, different numbers of hidden layers, which connect the input layers. The input layers are the features that you put in. I'll say in a moment what features I used. And then it tries to find patterns within there, and it gets trained on these data, and then it predicts what the input was in the output layer. So I took several eusocial and non-eusocial insect genomes, all of the available termite genomes that there were, and then the same number of eusocial hymenopteran genomes, and then some other genomes from within insects. And wanted to try and see if I could train an MLP to recognize sequences from eusocial and non-eusocial genomes. So I took several features, including hexamer scores. So a hexamer score is, so you look at, so I split it up in different sequences. First of all, each genome was split up into each exon went in there separately, each intron went in there separately, all of the flanking regions of each gene, so 10 kb in the 5' region, 10 kb in the 3' region, and everything in between, they were called intergenic sequences. And in each of these individual sequences, I had a look at the hexamer, so a sequence motif made up of 6 bases, every single possible combination of 6 bases, and then you count how many you observe in that sequence and how many there should be based on probabilities and the composition of that sequence. And then that gives you a score of the relative enrichment of a hexamer. I think there are 4,000 different hexamers. So there were 4,096 hexamer scores. We also put in the GC content and also the label of which genomic region that came from. And then I trained this machine on those, on a subset of the data, and then tested it on the test data set. And then it tried to categorize each sequence based on the features it was fed. Is this coming from a eusocial or a non-eusocial genome? And the first results are quite promising. If we first look at the flanking regions, 5' flanking region on the left and 3' flanking region second from the right, we have over 80% classification accuracy. That means whenever a sequence has come from the flanking regions, the machine managed to correctly classify it into either eusocial or non-eusocial. This came from a eusocial genome, this came from a non-eusocial genome. And importantly, it didn't manage to classify exon sequences. Now, if we had a really high accuracy level in exons, there'd be people shouting, oh, it's just recognizing phylogeny, it's just recognizing what a termite is and saying this is a termite and that's why I'm classifying it as eusocial. I don't believe it's doing that, because if that were the case, then the signals would be much stronger in exons. That's where you would more likely see a phylogenetic signal. So it's quite promising. This is, and it needs to be further developed. It's just initial findings. But moving on to the aspect of healthy aging in eusocial lineages, I started wondering whether there's any way of detecting specific signatures of healthy aging in eusocial lineages, and I did this by feeding in expression biases. So you have a look at the expression of a gene, the expression level of a gene in queens and in workers, and you compare that. And there's a measure which you can use there called fold change. It's basically just the expression in one caste divided by the expression level in the other caste, and then you take the log of that, so that you have this nice linear distribution. So in this case, this cartoon +10 would mean very strongly upregulated in queens and -10 strongly upregulated in workers, and vice versa, so down in queens. And I fed in these, so now it wasn't a classification anymore, it wasn't a binary classification, is it a eusocial or a non-eusocial genome? I only had one eusocial genome, and I was asking each gene, so now it's a regression exercise, right? And put it into the machine. Same sequence features as before, but this time with tetramer, so 4 base sequence patterns, and the different genomic regions, except for intergenics, or the flanking regions of the gene, the exons and the introns. It was trained on a subset of those genes, and then at the end, it's supposed to predict with unlabeled data, the test data that it's not seen yet, what is the relative expression of there in queens and in workers. And I did this with 2 data sets, one data set which we'd already developed for the termites, and one that was publicly available for naked mole rats. And I tested the accuracy of the model with something which is called the median absolute error. This, for each data point which goes in there, so each sequence, each sequence data point which goes in there, it predicts the expression value. That would be the predicted value here. And then you compare that to the true value, what it should have been, what it had been measured in experiments, and then you take the absolute difference of those two. So it's basically just the difference between prediction and true value, and then the median of all of those. So if it was working perfectly, you would have a median absolute error of 0. So the closer to 0, the better. And the median absolute error values at the moment for this machine are 0.6 and 0.2. To try and find out how good that really is, I estimated random error values. So if I just took the true values and randomized them and used them as the prediction and did that 1,000 times, I came up with these values. This is what we would expect if you just randomly put in values within the same range. So it's much, much lower than the random error. So it seems to be doing something quite well. But I'm also interested in age. So I also looked at age-biased expression. So how does expression differ in old individuals compared to young individuals, and try to predict that as well. And this is also predicted quite well compared to random values. So this is quite promising, and it needs to be developed further. It's still not dealing that well with extreme values, but in a small range, it seems to be able to predict the expression bias quite well. Okay, so it's possible with machine learning, I believe, to detect molecular signatures of healthy aging in eusocial genomes. Or what next? The first step is just to keep doing the genomics. I mean, we just need more data, and this is something I'm working on, keep developing more genomic resources. We have 3 projects ongoing at the moment. One of my collaborators is in the audience I saw today, Martin. So we're working on subsocial cockroaches, developing termite genomes, Ambrosia beetles, but also other social beetle genomes. These are either complete or being developed at the moment, and these will all be analyzed soon. In the future, I would like to sequence more mole rats and social mammal lineages, and then use the classic comparative genomic analysis to try and find convergent molecular signatures in each of these lineages, see if we see any patterns which are common across them, but then also use these results and these data to improve the machine learning algorithm. So train it to precisely predict sociality, caste and age-biased expression in all of the eusocial groups, and then use that to pinpoint the motives. which one's most informative in these decisions when the machine is managing to predict the sociality of the genome or the expression bias towards a certain age or a caste. Which structures, which signatures within the genome is it using to inform that decision? And then we go and have a look at them and manipulate them in eusocial and non-eusocial species and see if what kind of effect it has on longevity and fecundity. And this is at least my vision, and we have quite a few people who are on board who are willing to try and do these tests if and when we find some interesting motives. Right, with that, I'd like to thank you all for your attention and coming and listening to my talk, and all of these people, and for their hard work, and lots of other people that I work together with. I'm ashamed I don't have Martin on here, but he's included in the many more. Sorry, Martin. Lots and lots of people that I'm working together with, without whom this wouldn't be possible. Thank you for your attention, and I'll just leave it on a summary slide.
Yes, thank you very much, Mark. Great talk and interesting methods and results that you could show that you showed to us. I still stopped the sharing, though we can always go back to it. First of all, I would like to remind everyone, because I know not everyone can stay for the entire discussion here. Next week, there will be Peter Buston talking on social evolution in anemonefishes. So please, everybody come. And now we are going to, how do I unshare? And now we start the discussion. Like always, if you have a question, please write a question mark in the chat. When I call you up, please first introduce yourself. What's your name? You're coming from, what you work on quickly, and then ask your question. And for the people on YouTube, I see Adriana is checking this. If you have a question on YouTube, you can write it there in the chat, and we will transfer it here into the Zoom meeting. And the first question is coming by Raf, please. Let's see. Yeah.
Is that me?
Yes.
Yeah, can you hear me? Yeah, I am Raghavendra Kadakkar from the Indian Institute of Science. I work on primitively eusocial wasps. Thank you. I enjoyed this presentation very much, especially the machine learning part. And I look forward to what you are going to find with that technique. I have two questions. One is, would you expect big differences in the molecular signatures of healthy aging, as you call it, between facultatively eusocial species such as the wasps I study and more obligately eusocial like the termites and the ants and the honeybees? And my second question is, at least in the wasps that we study, when we keep them in the lab, their lifespans are much larger than in the field. So there's a big difference between physiological and ecological lifespans. Again, would you expect this similar molecular signatures or differences in the molecular signatures between well-protected lab populations and in the field, or would you at least predict if it has not been done yet?
Right, thank you for your questions. Yeah, that's something I would like to research. That's eventually I would like to expand the machine learning algorithm to include different levels of eusociality, and I'd be interested in seeing at what level we can detect a signature of eusociality at all, regardless of the healthy aging thing. My first impression is that the strongest signal, if only, is where you have true sterility, lifetime sterility. That's just first couple of analyses, like selection analyses on termites. But it would be great if we could see something in the facultatively eusocial insects. The question is, it's unlikely, but I'm hopeful. Okay. And the difference between protected and wild, it depends on what the causes are of the shorter life in the wild. If it's just general predation, and I think the comparison needs to be made between lab-raised species, and if you still find that these facultatively eusocial wasps are still longer lived than Tribolium, for example, it's not really comparable because they're really quite long lived. But other, I don't know if any other hymenopteran species are kept in the lab, solitary species like Nasonia, for example. I mean, if you still find that they're longer lived than them and they remain, the point is they remain fertile, no, until old age. Yeah, I'm not sure. I don't know about the details of different species, but it's something that I would feed into the model, right? So at the moment it's very, very basic, and it was just binary, eusocial, non-eusocial. I would try and feed in as much phenotypic data as possible and try and find out which phenotypic trait is most informative.
Yeah. Thank you.
Thank you.
Then the next question comes by David Queller.
Hi, I'm Dave Queller. I'm at Washington University in St. Louis, and I used to work on social insects, but now work on social amoebas. I particularly liked your result of sort of convergent result across eusocial groups where you showed elevated DNDS ratios for eusocial species compared to their non-social relatives, reflecting weaker selection, presumably due to the smaller effective population sizes. But it put me in mind of endosymbionts, these bacteria that live inside of other cells, and they often show that same pattern of elevated DNDS, partly because of reduced population size and partly because of lack of recombination. But they also show other signatures, including GC bias, codon bias, and often loss of genes, sometimes adaptive loss of genes, but sometimes non-adaptive. So do you see any of those signatures, a change in GCAT ratios, or increase in pseudogenes or anything like that? Or if you look for that?
Yeah, I mean, it's difficult with the termites. I mean, it's confounded with a change in ecology. And there's only one route to eusociality in the termites, and the wood feeders. I mean, we're hoping to be able to tease those things apart with the wood roach genomes that we've just sequenced. Right, so the sister group are also wood feeders, and then so they have a similar ecology to the termites, but they're not eusocial. And if we put them into the model, maybe we'll be able to tease these things apart. So at the moment it's difficult, and hopefully it'll be easier in the future. But I do see changes in GC content, but in different directions. I can't remember which way around it is, but in Blattodea, there's a strong change in GC content in Hymenoptera as well, between eusocial and non-eusocial, and it's the opposite direction. An endosymbiont.
Could I insert? That's thought to change because of mutational bias. It's also possible that mutational bias is different near different species.
Yeah,
but we don't know that probably.
Yeah. And I would expect to see changes in codon bias because that's related to selection efficiency. I've not tested that yet, but I expect to find a change in that. Gene loss, possibly. But with endosymbionts, isn't there also due to a redundancy that genes are lost, right? If it's an endosymbiont.
Sometimes it's just they don't need those genes in their host environment. But sometimes I think sometimes it is also due to just random mutations that get fixed, or at least it accelerates the loss of genes due to redundancy.
Yeah, once you have this dependency, that can happen. Yeah, we saw a reduction in proteome size in the termites compared to the cockroaches, but that's probably due to the specialization, the strong change from omnivores to these wood feeders. So hopefully we'll be able to find out whether we still find gene losses with an increase in eusociality. It's an interesting thing to look out for. At the moment, we don't know, but definitely changes in GC content and codon bias.
Thanks.
Thanks.
Thank you. Then we have a question by Eduardo.
Thank you, Mark. I think you all, I'm Eduardo Fernández Duque from Diego, one of the co-organizers of FINE. Thanks, Mark. I really enjoyed doing your readings. The readings you suggested, I struggle with them, but that's part of the fun of attending FINE. That it really gets me out of my...
I apologize.
No. I mean, it's the whole goal of getting yourself outside the comfort zone. So I really appreciate it. It was very good, very interesting. And it is because I mean, I want to start by acknowledging my most severe limitations in terms of genomics, in terms of machine learning, all these things that there are no doubts are changing everything we do. So what I have is probably an extremely basic question, but it can help me understand and imagine the way in which what we usually refer to as natural history and classic ethological studies, what place are they going to have as we move forward? As I was listening towards the last part of the talk with machine learning, it got me thinking about, we have now almost 30 years of GWAS studies. And I wanted you, if you could please comment on how the general approach to just looking at a huge amount of information and looking for correlations, should we really remind ourselves that we need to do it in parallel with maybe your classic behavioral genetics research, your natural history that will allow us to identify components of a potential signature that really are evolutionarily relevant, as opposed not, in parallel with? Because my understanding from GWAS studies is that for everything we expected them to do almost 30 years ago, it hasn't really played out as we thought it would. Not in terms of understanding the components of the signature that are evolutionarily relevant. So it's not a concrete question, but you have the expertise and I don't. How do you see that going in terms of what we can get from machine learning, what we can get for yet another humongous approach to looking at variation that we don't know whether it's predictive or explanatory, in trying to understand what is it really that the queen does that the worker doesn't, that is being acted on, evolutionarily speaking?
Yeah, great comments. Yeah, I agree. I mean, I think the only way forward is definitely working together, because and not being afraid to venture out into something, but also not being afraid to also support these ventures, right? So sometimes it's difficult for me to get support for this kind of venture because it's too exploratory. I mean, I've got good reasons why we should find a signature, but there are lots of very skeptical people out there, which is good. I mean, that's how science works, is skepsis. But I think the only way to do this is in teams, because I try to do it blindly, right? So I have these ideas of what I should be finding, and then I just explore, and then I find these things, and then we try and explain it, and then and I can't be an expert on all of the organisms, and that's why I have these really strong collaborations with lots of people who are absolute experts on different organisms, and together we try and understand things from both sides. So they also have some expectations of what they should see in their organism, and I have some expectations of what I expect to see in the genome, and then if we do find something, then we try and bring it together and understand it together. You had a wide-ranging question. I'm not sure if I really answered you sufficiently. It's just
you're sharing your thoughts, which is what I really wanted to hear, because you can think of this so much better than I can, and I hear you, because I feel the same. That I was listening to you, and I was thinking we need departments on aging, right? Not departments on humans, and I mean, it's really what you're saying, that we need to get in the same room, the same building, people day in, day out, which are, say, tackling aging.
exactly
in either the naked mole rats. No, yes, of course, you're addressing it well, and I thank you. I just find myself thinking, on the potential role going forward with everything that computers are going to do for us, of the actual identification of that behavior for which we can assess the functional value and hopefully find something similar across the taxa, which is what you're trying to do. Yeah. Thanks a lot.
Yeah, basically
Thanks
you need
a
to
lot.
have different people with different skills addressing a similar question.
Yeah. As I was telling you before you started the talk, I mean, I've been telling my colleagues who do human biology that they don't even know that this paradox that among the species you study, aging and reproduction really don't. I mean, you do not have the trade-offs that maybe people are talking about if you limit yourself to doing human biology. Thanks a lot, Mark. Look forward to talking to you later.
Thank you.
Okay, yeah, I have to say, like, for Eduardo, for me, it's quite complex. And like Mark says, we have different expertises and we can only learn from each other. And thus it's also especially good if the younger generation, the students, ask questions. And so the next question comes from Lindelani Makuya, or the group of students there.
Yeah, so hi everyone, I'm Lindelani Makuya, and I'm a research manager at the Sakhalin Karoo Research Station, as well as a PhD student working on the costs and benefits of solitary living. And these are the students that we work with here. So I'm the one who has a question. I just wanted to know, is it possible for climate change to have a significant impact on the way genes are expressed in the eusocial species, especially the termites, as we see how maybe they might be adversely affected by the changing climate?
Wow, great question. That's caught me off guard. Sounds like an awesome future PhD project. Okay, effect expression. I don't have an answer right now, I'm afraid. Maybe someone in the audience has a clever response to that great question. Yeah, I need to think about it. I mean, there is definitely an environmental influence on expression and caste determination in eusocial lineages, but how that is affected by weather, apart from just general survival of the colonies and the fitness of the colonies, I don't know, but I'm sure possibly maybe it's something that people are already looking into. But I'm sorry, I don't have a very clever answer to that.
No, that's okay. Thank you. I think it will be interesting to have students working on it in the future, then we can maybe get a clear answer on that. Thanks again for the.
Thank you.
Okay, then we have a second question by Rashavendra, please.
Mark, since you mentioned skepticism and said it was a good thing, I gathered the courage to ask you a skeptical question. Did you find anything that surprised you when you looked for these molecular signatures and the genomic signatures? Or did you at least say, I expected either A or B, and it just so happened that it's A and not B? Because the impression an outsider and a layman gets is that everything that you found is what we would expect. It's nice to know, but that's what we would expect. Did anything surprise you? Or did you say, was it this way? I thought it might be that way.
So, I mean, we're all guilty of storytelling in papers, right? So if you read the papers, it looks like everything was sort of what you expected. But no, I got surprised quite a lot during that study on the evolution of termites. So we were first looked at what had been found in Hymenoptera, and in all cases saw expansions of gene families related to development and communication and certain things. And then you look there and I just saw contractions everywhere, a reduction in everything, which was at first disappointing. And then we managed to find some kind of pattern in there, and then you try and understand it. So yeah, I'm constantly being surprised. And with the machine learning thing, I was surprised that it worked so well. I mean, I really, in this case, I really did have those expectations, and this is what I was hoping to have, and then it worked, and I was so surprised that I just kept looking for the error. I'm still looking now.
Mhm.
Okay, thank you. Then we have a question by Clara.
Hi, I'm Clara B. Jones, retired social evolution. I'd like to just comment on the climate change question. In Drosophila anyway, increased temperature increases mutation rate. So that's a possibility in which it could affect what's going on in the field anyway. But I have a question derived from EEB, and that would be, since longevity would be a mechanism to increase female fitness or lifetime reproductive success, wouldn't you expect, since some of the workers can become queens, wouldn't you expect a lot of competition between the females and some of the workers that can transition?
Competition for reproduction.
Yes, it would be costly to queens, I would think.
Yeah, but we don't see that. Are there any eusocial insect experts here? So there are people who study this in more detail who would be able to give a more informed answer to that. I mean, there are lots of developed theories on why there's less competition and why workers are willing to sacrifice their own reproduction. There we have an expert. Please go ahead, Raghavendra.
Yeah, I think we certainly see this very clearly in the so-called primitively eusocial species, where workers are totipotent. They can either be workers or become queens, and then the queens are replaced from time to time in the same colony. So there's clear opportunity for competition to happen. But the general sort of understanding, David Queller and John Strassman also have studied this extensively. The general understanding is that the workers try to trade off between getting indirect inclusive fitness by working for the queen's offspring and keeping a window of opportunity open for possible direct fitness in the future by their own reproduction. It is the sum of these two that really finally matters. And it probably doesn't matter how much you get from this and how much you get from that, but the total is what matters. So for some individuals, it might be worthwhile to revolt against the queen earlier than the others and get more of the direct and less of the indirect. And for others, it might be worthwhile maybe to get only indirect for their entire lifetime. So the sum of the direct and the indirect is really what matters. And how this is partitioned between the two halves depends very much on the individual physiology, on the ecological conditions, and on the social competition that exists in the environment. Of course, we cannot put numbers on this, but this is a general theoretical framework, and everything that we know seems to fit this framework.
Thank you. Thanks.
Okay,
And
thank you.
just to add to that, I mean, a lot of the first theories were based on Hymenoptera, where they're haplodiploid, and it was thought that that's the reason, because you're more closely related to your own siblings, you're more likely to care for them than to your offspring, right? But then the termites came along that are not haplodiploid. But then, so the theory was sort of broadened to say, okay, you don't have to be more closely related to the siblings, you have to be at least as closely related to your siblings, but you have to have some additional benefit for staying in the colony and helping out, which is shelter, for instance, and protection.
Yeah.
Okay, so if my observational skills are correct, we now have a question by Ching Yuki.
Hello, I'm Niño from I'm a PhD student from University of Strasbourg. I work on the personality of bush rat. If I understand it right, one of your slides, it says the selection works both directly on the queen, but also indirectly through the workers. So my question is, because I believe like the queen that is she doesn't have like a direct contact with the environment. Does this this mechanism makes the eusocial species genetically less flexible or sensitive to the environmental changes?
Question. I don't think so. I think there have been lots of work on this and lots of ideas of what should be happening. I mean, for instance, indirect selection should be weaker than direct selection. But then you have to also factor in the selection may be stronger on the workers, so it sort of cancels it out, and it's really difficult to find differences in selection between the castes. But whether they're right, the question was because the queen is not exposed as much to selection as the workers. It's not entirely true what I said there. I mean, the queens do go through a very strong initial selection phase, and that's when they leave their native nest and go and find a mate and found their nest. So there's already quite a strong selection filter there. But once they're in the nest, they need to produce healthy offspring and needs to have good control of the workers and so on. So the fitness of the queen is important, but also you can see the whole thing as a superorganism, right? So you've got the workers are the soma, and they're exposed to the environment, and they're the ones that need to perform well, and then any drop in fitness will have a strong effect on the functioning of the colony and also on the health of the queen. So if you look at it as a superorganism, selection should be just as efficient as individual organisms. But please, anyone step in and correct me if you feel strongly otherwise.
Thank you.
Okay, thank you. Then we have a question by Jonathan.
Hello, I'm Jonathan. I'm a first-year PhD student here at Yale. I have a question about your machine learning models. So for your first machine learning model that was predicting whether a species would be eusocial or not, you didn't specifically mention dividing into like a training and testing set, but you did for your later machine learning models. Is this just a problem with the amount of genetic like data that you have about, or like the number of species, or is it you did? And
good question. Yeah, I didn't go into the details. I cheated slightly, and that was just because I didn't have enough genomes, right? So I only had 5 termite genomes to work with, and ideally I would have an endless number of genomes, and some of the genomes I would just completely leave out. But I just kept repeating. I did separate out training and test data, but it was more of a validation. So there wasn't one separate chunk of the data which was completely left out till right at the end, and then to test it. So I kept doing leave in, leave out several times, going through and testing it over and training it over and over again with different separations of the data and different shuffling of the species combinations that I included.
thank you.
Yeah, so in the future, I'd like to have more data and then be more stringent with that.
Very well. Then I also have 1 or 2 very basic questions I don't understand so much. But first of all, I had to think about this question with climate change. and maybe what I say is stupid. But I mean, what is also very interesting with these termites and similar to the bush growers we work on are these extended phenotypes of these mounds they're building and these termite hills, which protect them against extreme environmental conditions. And this could mean that, I mean, they lived in this for millions of years. This could mean that they have reduced physiological and genomic flexibility to deal with something if something would happen. And the question is, could climate change lead to all of a sudden that they are exposed because of their extended phenotype? At some stage it breaks down. Let's say there's a heat wave, and when it goes above 45 degrees for 2 weeks, then there's too much hot air coming into these termite mounds, and might even be, like me here sitting in a cellar, be trapped inside and then lead to problems. I think it's a quite interesting question. For this to look at, one would have to look at these extended phenotypes as well. But with this, I come to one of my questions. I mean, you compare gene expression between queens of different ages from 3 to something like 20 months, and then the next step was from 3 months till 33 months, and then the next step was more than 20 years. So probably you have these big breeding colonies and then the younger ones. And you show these nice correlations, but is it possible that it's, I mean, 30 months is still probably quite young for a queen, and this 33 months old it was with the transposon study are very close to the very old ones, and it's instead that it's a line, it's actually only the very young ones, and then from 4 years on till 20 years, it's the same, like it's some ontogenetic during early development, for which if you get 20 years, then 3 years is only a part of it, and then it rather looks like that. And it's only because you cannot have queens with 5 years, 10 years, 15 years in between.
Yeah, the arrow was a bit misleading. I was not trying to say that there was a correlation. But it's true. It's interesting that it's already well regulated in the 31 or 33-month-old queens. And that's something we need to try and understand better. This is also when they become physogastric, so they have this huge enlarged abdomen when they start turning into these enormous egg sacs, basically. And it could have something to do with that. So they already have that at that stage. So they've gone through this redifferentiation of the fat cells at that stage and don't change much over those 20 years.
So might the driver rather be the changes in body mass than age? Because in the same graph, you have these interesting comparison with 20-year-old kings, and they are just not good in controlling these transposons. They're like young queens, but from what I know, the kings are still very small. They don't have this big transition in getting these huge animals.
The point is that it's portions, right? It's the proportion of transposable elements within mRNA. The absolute level remains relatively stable. So the point is, as they increase expression, they manage to control the TEs really well. We don't see that in that graph very well. So there's like an 8-fold increase in overall gene expression in the 20-year-olds compared to the 31-month-olds. And that's why compared to the young ones, sorry. Right, so there's this massive increase in expression, and that's why the proportion of TEs gets really, really low.
Okay, I'm not sure if I fully understood this. How do you explain the difference between the kings and the queens?
I can't.
The old queens.
Yeah, we don't have an answer to that.
Okay. But is it correct what I said, that the queens have this individual development of not only getting old but very big, while the kings don't have this? They don't get very big.
Yeah.
So
that's true. Yeah,
it could also be related to this, and of course, when you have a much bigger soma, you have a much higher.
Yeah, I mean, it's a bit more costly for the queen than the king. The king doesn't need to do any of the storage that the queens do.
Okay. Then just out of interest, and I mean, you mentioned you have to work together with other people, of course, which is important and good. With the naked mole rats and the social mammals, with whom are you working there together? Are you just getting data from published databases, or?
Yeah, yeah, this is brand new. I don't have any collaborators there yet. I'm putting out my feelers at the moment, and I've just been working with published data.
Oh, okay. But do you have funding to develop this project bigger? Do you looking for students, postdocs, or what?
It's in the pipeline. I don't have it yet, but I have proposals and just need to get them funded. I'm still developing them, right? So trying to get these initial results a bit better before I resubmit some proposals. But yeah, I will be looking for PhD students or a postdoc possibly soon.
Okay. Then my last question that might also rather mirror my limited understanding of machine learning, where you had this interesting comparison between healthy and unhealthy aging. Would such an approach also be useful to understand better the difference in humans if you would have, let's say, two cohorts, healthy old people, unhealthy old people, and feed the machine learning with finding out why do some of us, or what is the genomic differences of the humans that get healthy old and the ones that develop lots of problems early, or would it
definitely, yeah, that's a good idea. Yeah, it's a good idea. I'm not sure if people are already doing that. It makes a lot of sense.
I was just trying, I mean, we're always forced to make our research applicable, and it was just coming to my mind when looking at different old people. There's a huge, some of them are always healthy, and some they start getting old and sick with 40, 50 or something like this. I think it's always amazing.
I'm going to steal your idea.
You brought the idea into my mind, so it's yours. But okay. Thanks a lot. But if there's no more questions, I might still ask another question for totally showing my lack of understanding. But I thought this might be important also in the field of evolutionary medicine, where we just wrote some reviews and I didn't see this. Can you explain to me once more the selection shadow? Is here the idea that some genes are not under selection because the animals normally don't get as old, that this would be relevant, or what was the idea here of selection shadow?
So it's the evolutionary theory it's one of the evolutionary theories of aging, why we age, why there's a breakdown in different functions. And one idea is that selection isn't efficient enough to maintain the functioning of genes which are important for survival at old age. And that's due to a declining number of individuals that survive. So a gene which is really important for your survival at old age is only going to be expressed in much fewer individuals. So the affected population size is lower, and that decreases selection efficiency. So it's like a trade-off between young and old, and there's
Carsten, you're muted.
Yes, I'm muted. So thank you very much. I mean, I just looked on also on YouTube, but I anyway checked it. There's not any more questions in the chat or here. So I'm for now stopping the live screen on